Hello, myself Nidhi Trivedi. I am associated with Microbiology Department of Sri Maniklal M. Patel Institute of Sciences and Research, Gandhinagar. In this video, I am going to talk about polymerase chain reaction or PCR. So, suppose we have a certain segment of DNA molecule that we want to amplify. To amplify means to make many identical copies of that DNA. Now, the question is, how can we go about amplifying that gene of interest? So, one of the way is to take that gene, integrate that gene into a bacterial plasmid, place that recombinant plasmid into a bacterial cell, and then allow that bacterial cell to divide many times. Eventually, our gene of interest will be amplified. But the problem with this method is, it's not only time consuming, not only it is ineffective, but it also limits the size of the gene that we can actually use. So, a much more effective, efficient as well as accurate method for the amplification of DNA is the polymerase chain reaction or PCR. So, it is a technique that allows us to make multiple copies of a segment of DNA of interest, generating a large amount of copies such as millions or billions of copies from a small initial sample. As it is an in vitro DNA synthesis method, it can be performed by hand or it can even be performed in a machine called as thermal cycler or thermocycler. So, this technique was developed by an American biochemist named Kadi at California in 1985. He even shared Nobel Prize in Chemistry in the year 1993. Now, let us see what are the ingredients required for this reaction. So, we have four important ingredients for this reaction. The very first ingredient is the template DNA. That's a target DNA molecule that contains that sequence or a gene that we wish to amplify. The second important ingredient is a pair of primers. So, what are primer? It is relatively short, single-stranded sequence of DNA and oligonucleotide of about 20 to 30 nucleotides long. It is useful for the initiation of DNA synthesis. The third important ingredient is thermostable DNA polymerase because the reaction is carried out at relatively high temperature, and one of the thermostable DNA polymerase used in PCR is Taq polymerase, which is obtained from a thermophilic bacterium named Thermus aquaticus. The fourth important ingredient is four different types of deoxynucleoside triphosphates. Now, let us see what are its steps. So, there are three steps within a single cycle of PCR, where step number 1, we call it as DNA strand separation or denaturation of DNA. Step number 2 is hybridization of primer onto that DNA on which we want to amplify. That is called as primer annealing. Step number 3 is DNA synthesis process, which is called as primer extension or polymerization process. Now, let us see all these three steps in detail. Here comes the step number 1. As we want to separate these two strands, so the temperature is increased till 95 degrees Celsius for 15 seconds. So, what will happen with increasing temperature? So, it will break the hydrogen bonds between those two strands of DNA and it ultimately leads to denaturation of DNA. This process is called as melting of target DNA after separation of each strand acts as a template for DNA synthesis. Now, once we have separated the two strands, we begin to decrease the temperature. So, the temperature is held for about 68 degrees Celsius for 60 seconds. The reason the temperature is lowered because at this temperature DNA primers will bind. The second step is the annealing of two oligonucleotide primers to the denatured DNA strands. Since nucleotide sequence of each of oligonucleotide primer is complementary to 3' end of single-stranded template, the primer anneal, that is hybridizes the each template. The primers are added in excess amount and the temperature is lowered to about 68 degrees Celsius for 60 seconds. One of the primer bind at one end, the another binds at the other end of the DNA. The reason that they bind at 3' end is because the DNA polymerase can synthesize from 5' to 3' direction. Now, in the third step, again the temperature is increased to about 72 degrees Celsius and thermostable DNA polymerases are added along with deoxynucleoside triphosphates. The DNA polymerase accelerates the polymerization process of primers and therefore extends the primers, resulting in synthesis of copies of target DNA sequence. Now here is the diagrammatic representation of all these three steps in detail. There is a single cycle of PCR in detail. As we just discussed, the first step of PCR, that is denaturation. Here the strand separation of template DNA occurs where the temperature is increased to about 95 degrees Celsius and it is held to 15 seconds. Here the hydrogen bonds between the DNA breaks down and ultimately it leads to separation of these two strands. Further, the next step, there is incorporation of primers into it. Here, the primers will anneal to 3' end of both the strands where the temperature is lowered down to 68 degrees Celsius and it is held for 60 seconds. The third step, which is polymerization step, where there is incorporation of deoxynucleotide triphosphates along with that of the Taq polymerase and magnesium ions into it, where the temperature is increased to 72 degrees Celsius and here the Taq polymerase will extend this primer and ultimately gives us two DNA strands from a single DNA molecule. So, one cycle of PCR is completed, giving two copies of the desired DNA. Once a new DNA strand made via the first PCR cycle, it serves as a template molecule in subsequent cycles. The number of double-stranded target molecules obtained after 30 cycles yields us millions of copies of our desired DNA target. Now let us see what are the applications of PCR. So, one of the first important application of PCR is diagnosis of pathogens. So, several pathogens are known that grow very slowly. Therefore, their cells are found less in number in the infected cells or tissues. It is difficult to culture them on artificial medium. Hence, for their diagnosis, PCR-based assays have been developed. Next important application is diagnosis of specific mutation. So, the presence of faulty DNA sequence can be detected before establishment of disease by using PCR, sickle cell anemia, phenylketonuria as well as muscle dystrophy can also be detected. Next important application is prenatal diagnosis. Yes, it is useful in prenatal diagnosis of several genetic diseases as well. Next important application is DNA fingerprinting. The DNA fingerprinting is more successfully used in forensic sciences to search out criminals, rapists as well as solving several disputed cases. Also for the uniting of lost children to their parents or relatives by confirming their identity. Here also PCR plays a very important role because it is done through making a link between DNA recovered from samples of blood, hairs at the spot of the crime and the DNA of suspected individuals or between child and his or her parents or relatives. Next important application is in the field of research. So, DNA fingerprinting of new microorganisms isolated from various extreme environment is also carried out to confirm their identity by comparing with the DNA sequences of known microorganisms. Their DNA as well as RNA can also be amplified and it is also useful to determine the orientation and location or restriction fragments relative to one another. Next important application is in molecular archaeology. So, PCR has been used to clone the DNA fragments from the mummified remains of humans and extinct animals such as dinosaurs from the remains of ancient animals as well. And also nowadays, DNA from buried humans has been amplified and used to trace the human migration that occurred in ancient time. Another important application is for the diagnosis of plant pathogens. A large number of plant pathogens in various host or environmental samples are detected by using PCR technique as well. So, this was all about PCR. Thank you for watching this video.